Right. So I'm pretty sure that the abstract is due on February 15th, which is not this Friday, but next Friday. So if you're thinking that you might want to go to the UMass Research Conference, then we need to pound out an abstract next week, right? So um, I have a student in my Bio 2 class that is interested in doing that. So on Thursday at our Bio 2 lab with the library, I'm going to gather some materials with Tim. We're going to give him some abstract writing materials, and I'll post those on our Moodle page. I haven't turned on our Moodle page yet, but I'll turn on our Moodle page and I'll put some resources up there so that if you guys think you might want to do the UMass Research Conference, you can at least look at some of the abstract writing guidelines and think about what you would have to put together. And then we can spend a little bit of time working on that next week if we need to. Our plan for today is to talk about types of ecological study and what we can do, and spend a little time talking about what kind of things you guys might want to do for your projects and kick around ideas a little bit and see how long that takes. And then depending on how long that takes, we may also talk about reading primary literature today and do a little activity where we actually read a primary paper and think about getting information from it. And I want to talk to you about where resources are available for you thinking about starting your projects. So did everybody read that Designing an Ecological Study reading that I gave you? Yes. One of you is going to nod. Everybody's like, did I? I carried it around for a week. I didn't actually open it. So there's some pieces of this that are not particularly useful. There are other pieces that are a little bit useful. And so it's sort of just starting to talk about some pieces of how we can design an ecological study. So I want to talk a little bit about types of research and the types of research we might do in here. So I'm going to talk about all of the types of research kind of largely and colloquially, and then talk specifically about the kind of projects we can do. So one of the things that I think happens when we start a project like a research project for this class or for ecology um, is that you guys start thinking about what research should be. And you're like, oh, I want to do this really big thing and I want to compare these different sites. And sometimes your eyes are bigger than is possible because you have to remember that we are not researchers out in the world functioning as ecological researchers where we have multiple summers of field seasons to do our data collection. We have between now and the middle of March or May, right? We pretty much have from spring break to May 1st to gather our data. So mid-March to the beginning of May or something like that. We have a really small window. So what we can do in that really small window is also going to be pretty small ecologically. So I'm going to talk about types of ecological studies so you know what's out there, but we are not going to do a controlled manipulative experiment across multiple sites, right? It's not going to happen. We don't have the time or the resources to do that, okay? So I put this picture up here just to show you a big, large scale landscape level project. And I actually don't know what this project is. I pulled this from, yeah, this is from the University of Minnesota project. But there are a lot of projects that look like this, where basically what they've done is they've taken this really big field and they've divided it up into a number of small plots, right? And different plots are probably experiencing different treatments. Maybe that's different levels of supplements for plant growth, like maybe we're feeding it different nutrients. And so I know someone who did a study that looked a lot like this, where they were actually looking at small mammal use of different squares. And so they had small squares and medium squares and big squares. And they were looking at how animals use that space. And it turned out that the amount of edge relative to interior habitat impacted how big the population could be. The animals were more scared on the edge. And so a bigger block could have higher densities of rodents than a smaller block, right? So this is a big manipulative experiment. We've mowed a field into experimental units and we are manipulating them. This is the kind of thing that we could never do in the confines of this class, right? But a lot of big ecological projects try to control nature in some sort of way so we can actually ask really specific questions. One of the biggest problems with ecological research from a statistical analysis standpoint and from a strong sort of data standpoint is that it's really hard to manipulate the natural world and ask questions about the natural world without thinking that your answers are a result of your manipulation, right? It's hard to study the natural world without disturbing it. And so when you go out and you want to look at how organisms are interacting with each other in space, your presence alone impacts that system and changes things about how those organisms are interacting. And so ecological research is tricky, and it's often really time intensive and or money intensive and can be really expensive because of the scale. So when we're thinking about our projects, we're going to really try to find something very small scale and keep our, expectations low and reasonable for what's manageable in this class, while introducing ourselves to the materials and the methods of the process. Okay, so ecological research comes in a lot of different flavors. We can have qualitative research and we can have quantitative research. We're going to talk about both of those in a little bit of detail. We might have observational studies where we're primarily just going out into the natural world and recording what we can learn about it by watching it or looking at it and maybe taking some measurements of it from a distance, as opposed to an experimental study where we might set up some kind of manipulation or try to create an environment within the natural world that we can control or that we can match variables between one location and another location. And then we have field studies and we have simulated environment studies. We can do ecology in the greenhouse. We can do ecology in a laboratory setting. It's harder. We're looking at a subset of the entire ecosystem when we're doing that, but it's possible. I was really thrilled last week in Bio 2. Tim said that if you do biology studies, you go to the field. And it was just like, it tickled me so much because like a lot of people think if you do biology studies, you go to the lab, right? Like just training him to be an ecologist. I'm taking an ecology bent here, obviously, right? There are lots of studies you would do in a greenhouse or in a lab if you wanted to really control things and manipulate things. But often in ecology, we want it to be as natural as possible. We want it to be as much like the natural world as it can be. And so instead of trying to control things and manipulate things, we're actually trying to simulate nature as much as possible. So we might create, large tanks that we use as simulated lakes and try to set up a fake ecosystem and observe it. Okay, I feel like I just need, this is actually recording, right? Yeah. This panic moment that I'd canceled it. Sorry. Okay, so let's think about qualitative studies. What does the word qualitative mean? Not numerical. Quantitative is numerical. Qualitative is descriptive. So like a verbal description. So if I wanted to say something qualitative about myself, I might say I'm of average height and I guess my hair's brunette, right? It was always blonde for so long, but I don't think I'm a blonde anymore. Whereas if I wanted to be qualitative about it, I would want to say I'm 5 feet 4 inches. And I could actually measure my hair color with paint swatches or something, right? Like you'd have a board of paint swatches and say, well, she's a, brown 18 or whatever the number is that you delineate, right? So there's like two different ways of thinking about observing the world. And so even, in a very observational study, we could be doing things like quantifying hair color or feather color if we wanted to, right? It doesn't have to be just a description. But qualitative studies tend to include a lot of observations and descriptions. I did. It's okay. Um, I was asking you to answer it without looking at the slide. Um, it doesn't necessarily, by saying that the study is qualitative, like calling my hair brown is a qualitative description, right? But a qualitative study doesn't have to be absent of any numbers, okay? You might do a qualitative study of GCC's forest, in which you go out and observe and describe the forest. And you might say, based on my observations, this forest is make up random stuff, so don't believe anything I say. 40% maple and 10% oak and 10% beech or whatever, right? You might describe the tree makeup of the forest. You might describe the soil type and the soil pH of our forest. You might describe the understory. You could talk about what the sunlight penetration through the overstory is, like how much sunlight hits the ground on average throughout our swath of forest, right? A lot of that would be really numerical, 10% of all light reaches the forest floor or whatever, but still the study itself is qualitative, it's descriptive. It's based on observations. And you're telling us, like, what is this forest like? Okay. Often qualitative studies are exploratory. If I want to compare GCC's forest to, say, the forest out in Northfield at Masson Ridge, since this is another site we have the potential to work at, before I can compare them, I have to qualitatively assess both of them, right? I might do that at the same time, but I can't compare them without describing them both, right? You have to describe first. So often the qualitative study is a critical first step. Like before we can ask a strong research question about our environment, we need to know what it is, right? If we wanted to study the bears in the GCC forest, right? First we have to know, are there any bears in the forest, right? First things first, we can't try to guess how many there are until we know if they're there, right? What other large mammals are in the forest? Maybe we want to do a survey first. And so it's often a first step to get to know the habitat. So last year, this class, several students went in, looked at the wetland on campus, and we pretty much just captured everything we could and listed what species are here. We didn't even try to figure out how many of any of those species were there, just like who lives in this wetland, just trying to describe it, okay, get a basic understanding of it. So this provides the knowledge and insight that's needed for further study, right? We can't go to the next step until we do the groundwork. And so qualitative studies are often laying the groundwork for further work. When you're doing this kind of a study, even if you're collecting a lot of numerical data on your site, you're usually kind of just describing one place or one organism or one piece. You're not doing a repeated measurement across multiple locations. And so the scope is limited to the place you're actually doing your observations and descriptions. You can't go out and observe and describe GCC's forest and then say all forests in Massachusetts are 40% maple, right? It's not extrapolatable. That's not a word, is it? To bigger scales, right? It's that you know something about the scope at which you worked, but you don't know something about the world at large until you compare it to other sites. Does that make sense? Okay. So surveys or observational studies, some examples might be looking at spatial or temporal differences in a particular variable. That sounds pretty quantitative in some ways, but that's still a qualitative and descriptive study. So maybe you want to see if different parts of GCC's forest have different makeup of tree species or different soil profiles or soil pHs, right? Maybe you want to go back year after year and see if things are changing. So maybe you study, I had an example for Taylor, the meadow every spring for 10 springs, and you kind of describe the changes over time, right? You could also do, like I just described in the wetland, a taxonomic exploration or cataloging of the diversity. What kind of plants are here? What kind of animals are here? What are the basic physical environmental characteristics? What's the underlying geology? What's the soil structure and soil chemistry? What is the atmospheric quality or average temperature or relative humidity, right? Beginning to describe a space. So anything along those lines would all be sort of a survey or observational study and considered more of a qualitative study, where you're just giving a general description of a place or an organism. Does that make sense? In my opinion, this is where we're at in our class, right? The vast majority of the projects that we can reasonably do in our limited time frame with our limited budget fall into this kind of a category. We can describe, we can observe, we can catalog species, and we can quantify diversity, especially of things like trees that don't move. But we're going to be limited in how much comparison we can do. Okay. So if we compare that with a quantitative study usually focuses on taking objective measurements of predefined variables. So I'm going to take these objective measurements, and maybe I want to look at, now I'm digging deep in my forest example, tree height by species at 3 different forest sites, right? So I'm going to record the tree height by species, which we could do at any one of those sites, but now I'm doing it at multiple sites, and I'm going to look and see if there's a statistical difference between sites. Are trees taller in Northfield than they are in Greenfield? Not sure that's a useful research question, but I'm on the spot here, right? And so here we're taking a predetermined set of measurements with the expectation that we're going to do statistical analyses, that we're going to compare them, right? So one group that took this class last semester actually did a study like this, and they took all of our salamander capture data from here, and they looked over all of the captures we had done in 2 years at the temperature and soil moisture calculation relative to number of individuals captured. And so they were looking to see if there was a relationship between soil moisture or temperature and the number of individuals captured. So they performed a linear regression and statistically compared the data and looked for an outcome. Of course, that project would be much stronger if they did that not just with data from GCC, but from all over Massachusetts or from all over the range of the animal. Okay. The way you expand your scope is to expand where you compare sites from. So if I only study redback salamanders here on GCC's campus, then when I make a generalization from that study, my generalization is only good for GCC's forest and the salamanders that live in that forest, right? If I collect data from here, and I collect some data from Fitchburg and from out at Wendell State Forest, and some data from over on the coast, and some data from up in Turner's Falls, and all the places I know that we have this project running, then I can say something on average about terrestrial salamanders in the state of Massachusetts, right? What the SparkNet project is doing is collecting data all over the entire range of the species. So we go down to Virginia and Maryland and up to Michigan and the lower Canada. And so if we compare across all of those sites, then we can say this species on average does this. Everywhere that it's found, we have some general trends. And so when you think about the scale that you're doing your study at, the bigger your comparison level is, then the more generalizations you can make about the thing you're studying. When you're only studying at one place, then you can say, well, I know something about this place. Does that make sense? Okay. All right. One of the things that we can do if we're doing a very quantitative approach is to do a manipulative experiment. And so here we might actually ask about the effect of one factor on another variable. So how does factor X affect variable y. Here we're trying to look at hypotheses of cause and effect. If I manipulate this variable, how is something going to respond? And so a really simple example of this might be to think about fertilizing plants, right? If I add fertilizer, then the plant will grow taller, right? If I fertilize the forest, will I get bigger ferns, right? So how does manipulating a particular variable impact the response you see in an organism? I am going to post these slides for you guys. I feel like you're writing frantically over there. These are really text-heavy slides, which is atypical for me. We're not having any exams in this class. Right? This is your background information for thinking about designing your project. Okay, I can't even, I'm like, is it really 3 o'clock already? It is. Where does the day go? It's gone. Okay, so if we're going to manipulate something experimentally, we have two types of variables that we're considering: the independent or predictor variable and the dependent or response variable. You have worked with these variables in other classes. You just maybe haven't thought about them in this language. Okay, so imagine that I'm going to make a graph of, oh, let's say, what's a good ecological example? We're just going to use a human example because it's the easiest one I can think to come to my head. Let's pick child height over age, right? So this isn't actually an experiment. This is an observational study, but it's really easy to think about how these two things go together. As the child ages from 1 to 10 to 20 years of age, presumably height is going to grow kind of probably really pretty quickly. I don't even know what this figure looks like. And then they're going to reach their adult height and level off, right? So the independent or predictor variable is down here on the x-axis. So in this example, that's age. And then child height that we're measuring is going to be the response. As the child ages, their height is going to respond. Okay. Usually our independent variables or predictor variables are categorical. So things like species or sex or age or time or space, between plots, over years, over a lifetime, across different but similar species. Okay, usually that x-axis, our independent variable, is something categorical, maybe qualitative, maybe quantitative, but definitely a category, like a bin we can put things in. Organisms that live at this location. The dependent or response variable, the y-axis variable, is usually a quantitative measure. So if I wanted to look at species diversity in 2 Massachusetts forests, and I picked Northfield and GCC, and at these locations I want to know number of individual bears per acre. And so I might find that at this predictor, site, category, location, I have 2 bears per acre. That's really high. I have half of a bear per acre. And at Northfield, get something biologically appropriate. I have 3/4 of a bear per acre. That's still too high. We really needed to have done this by 10 acres. Bears take a ton of space. But just making some things up out of my head, right? Your y-axis is usually a quantitative measure. It's a count, it's a height, it's a number, it's a mass or a weight. And so our category, if we think about something like Jeremy's interested in soil chemistry, maybe our x-axis is pH and our y-axis is overstory height. Something soil pH impacts tree height, adult tree height or something. It's making things up, right? But our response variable is usually really quantitative. It's the number of individuals, it's the frequency of which a behavior occurs, it's height, it's weight. It's something that you measure, and that measurable response is predicted by the category, okay? So we're going to use these dependent or response variables in our independent and predictor variables. You can choose which one of these makes the most sense to you colloquially. So an independent variable is that category, and the dependent variable, it depends on the category. How many bears do we have? Well, it depends on the site, right? You can think the same way with the predictor and the response, right? How many bears do I have in response to which site I'm at? Okay, the predictor predicts the response. The independent variable is related to the dependent variable, which varies based on site or time or space. Does that make sense? Okay. So even though we might not necessarily do a whole lot of like experiments at which we would be doing this across an experiment, these terms are also important anytime you're making a figure. So if you're thinking about how am I going to represent my data, well, you might want to think about what are your categories that you want to represent? What are your x-axis variables? And then how do you want to display the data in response to those? Okay, so these terms are relevant regardless. It depends on how much money we can scare up for trail cams. Bear traps. Hair traps. Hair traps. I said bear traps, I meant hair traps. It's really hard to get a bear to go into a box. But they'll come to trail. They'll come to trail cams for your day-old donuts, though.
They will.
You can lure them in with food. Yeah.
That's when you're really good.
Yeah. We're not going to shoot our study organisms.
Also, like I put on my bait piles, I say these are for squirrels. So for squirrels, use only and leave here, not eating.
Okay. Another type of quantitative study is a manipulative experiment. And so I actually put an example up here to think about. So we could conduct an experiment to answer the question, does horned lizard predation impact ant population density? Does everybody know what a horned lizard looks like? How did I not put a picture in here? I think we're going to take a pause, even though Riley's not in this class. I forget, I was doing something yesterday. I was like, we need a video of this. And he's like, we'll wait. Go get a video. Horned lizard. I feel like we should be able to know what these guys look like. Have you guys seen these before? There's a whole global species group of horned lizards. They live in arid desert environments. They've got these spiky heads. They're sometimes referred to as horny toads. You didn't know there was something different? Yeah, yeah. So these guys they're technically called horned lizards. Some of them can actually shoot blood out of their eyes and do other crazy things. I think that the horny toad nickname comes from the horny structures on the head and the fact that they look kind of like a squat toad because they're kind of low to the ground and fat bellied. They look a little bit like a toad. Most of them are phenomenal consumers of ants. The ant walks by and they lick them up. And so they eat really high numbers of ants for their body, over time. So my question here is, does horned lizard predation impact ant population density? So how could we answer that question, right? We could answer that question in a lot of different ways. We could go out and start by just doing an observational study, like how big is the horned lizard population? How big is the ant population? Is there some sort of correlation? Do you have more ants when you have fewer lizards or not, right? A more simple descriptive type study. If we wanted to do a manipulative experiment, we would want to really set up our hypotheses and we would create some sort of environment in which we can actually ask this question in a manipulative way. Okay, so
put more horny lizards in one area than another area.
Yep, we probably would involve building enclosures. Yep, in order to contain them, and then experimentally manipulate population sizes. So before we get to that part, I was going to go through a couple of other terms. So I don't think that I have talked in many of my classes about the null hypothesis. We just talked about this a little bit in Bio 2 today, right? In statistics, you talk about it extensively in statistics, right? The null hypothesis is essentially the hypothesis that there's no significant difference between specified populations. So any difference that exists is due to chance or some sort of sampling or experimental error, as opposed to a biological mechanism. So if we went out in our observational study and we wanted to look for a relationship between ant population density and lizard population density, and we measured them, our null hypothesis would be that ant population density is not related to rate of lizard predation. Regardless of how many lizards we have, ant population is going to be the same. Okay, there's some sort of hypothesis of no difference, right? We usually use that null hypothesis, the no effect hypothesis, to build our alternate hypotheses, right? And so our alternate hypotheses might be that ant population density drives lizard population, or lizard predation drives ant population. We have something much more specific, okay?
You can think two sides of that. Increased predation trigger an increased reproductively.
Right. Ants might respond to increased predation by making more ants.
Like we see with
yep. Like you see with the coyotes. Yep. Totally possible. This should say H1. This is a typo. So an alternate hypothesis, hypothesis 1, the null hypothesis is always H0. So that's a copy and paste issue. Sorry. So an alternate hypothesis might be ant population density will be lowest when lizard predation is highest. That's kind of an intuitive hypothesis, right? I could make H2 and put Sean's hypothesis up there, right? Ant population density is highest when lizard predation is highest because they have some sort of reproductive response, right? And so we could list out all of our possible alternate hypotheses. And this is a good task for any of your research questions. And usually, when we are embarking on research, we like to use what's known as the multiple working hypotheses method. So we would, once we've determined our research question, we would write out the null hypothesis. What does it mean if there's no effect, right? And then we would write out all of our multiple working hypotheses. For this particular random example about ant population density, we've just come up with two alternative hypotheses. Either ant population density is lowest when there is a high number of predators, or it's highest when there's a high number of predators, right? We could list out all of our possible expected outcomes. And then when we gather our data, we systematically try to reject those alternate hypotheses. The data don't support this hypothesis at all. I'm going to put a line through it. What about the next one? Okay. So if you imagine trying to do an experiment to really get at this, right? To actually manipulate, it's really hard to do. Because if we really want to know, we're going to have to somehow add more lizards to a population and look at the change in ants. And in order to do that, we probably have to make it so that the ants can't escape, right? So we have to pick some sort of realistic size habitat, like what is the home range of a given lizard, and what is the home range of an ant colony, and kind of use that to decide, well, how big should our enclosure be? And if we want to really get at this, we need more than one replicate. We probably need more like 10 replicates. And now we're building 10 large enclosures out in the desert, and we're trying to gather data on all of them and compare, right? So if we wanted to do an observational study, we could just go out and look and see like how many ants are there and how many lizards are there. But then we're limited to looking for correlations. We can't really say anything about causation, right? If we want to really get at causation, we have to try to actually look at the response to a change. So generally speaking, in ecology, manipulative experiments are really valuable for the data they provide, but they're really hard to do. They're really expensive. They take a lot of manpower, like a lot of human bodies have to be out collecting the data. This is something you would often have to rely on getting a big grant for, having a whole team of people ready to do in order to do a manipulative experiment. It's really hard to have many replicates. And so you could do this manipulative experiment for lizards and ants, right? And that's possible. Maybe do it for rodents. But imagine trying to do a manipulative experiment where you are like manipulating the space that a bear uses, right? How big would your enclosure have to be if you wanted to not let a bear leave your enclosure? Right? It's huge. So it's hard to manipulate the environment
without affecting their behavior.
Yeah. Right.
Captive bears going to react.
It's going to act differently than a free-ranging one. Right. And so if you wanted to manipulate something like berry availability within a bear's home range and look at their diet in response to that, you'd want them to not be able to leave, but then you'd also like have to go plant berry bushes or something like that. It gets really big over time.
Reproductive rates of black bears in Maine, they kind of follow along the mass production of.
Oh, right, right. How much beech tree mass there was relative to bear reproduction. An interesting, like not natural experiment, but big scale manipulation. A friend of mine did this study in the 90s because when they take black rhinoceroses are horribly poached. Populations are in huge decline. When they bring them into the protected enclosure, they don't reproduce. If they do reproduce, their offspring don't survive well, and their health declines. And so they're like, what's going on? Like, how do we keep these animals healthy while we're trying to protect them? So her hypothesis was that there's something that they're eating in their natural diet that they're not getting a nutrient that they're not getting when they're in captivity in these big open enclosures. So they went out with huge dump trucks and gathered literally thousands of pounds of vegetation every couple days with this massive team of people. And then they hung the vegetation in the enclosure, and somebody sat there and watched and recorded how many bites the rhino took. They recorded the biomass left at the end of the day, like how much did it eat. And it turned out that there are some things that are missing in a captive diet. And if you put those vitamins back in, they do better. And so think about like trying to answer some of these questions, the amount of energy. Right. White rhino do much better in captivity. Yeah, that's a subspecies.
Oh,
Yeah, yeah. They but I mean like rhinos are horribly persecuted for poaching. Like there's just it's hard to keep up with it. But in general, the southern white rhino breeds really well in captivity. Um,
rhinos and gorillas.
yeah, yeah.
Stupid gorilla hands.
Right. Horns. The most trafficked animal right now is the pangolin. Pangolin. Pangolin that's like a little anteater looking armadillo-like creature. It's a status symbol soup, and so the bodies are actually infants are actually put whole in broth as a soup, and then their scales are also used like rhino horn for traditional medicine. But yeah, there's lots of we've really gone down the rabbit hole there. But yeah, there's lots of lots of experiments to try to manipulate these organisms' natural environments also. Like how do we keep them healthy in captivity to try to protect them? Okay. To come back to, I just like, I don't, my brain took a left turn there. Come back to ecological research. We also do a lot in ecology, what's called a natural experiment. And we read about these in our ecology books. We talk about them. Natural experiments are often not actually an experiment at all. It's really an observational study. The experiment part is like actually in the name, nature did an experiment, right? And so this takes advantage of naturally occurring variation. Um, so we could find locations with high lizard populations, and we could find locations that are really similar in their vegetation and their geographic structure and their underlying substrate, but that have lower lizard populations, and then look at ant numbers and see if there's a relationship, right? Again, we're stuck to a correlative relationship, but we could do that. You might also look at an area before and after an event. And so I probably mentioned this before. I had a friend who was studying nutrient cycling in Toolik Lake up in far up in the northern reaches of Alaska. And he had been studying this site for a couple of years. And he was studying it when the first tundra fire occurred and burned the environment around his lake, right? So we had all he had all this data, and then the fire occurred, and he could look at the nutrient loads post-fire too. And so that's a natural experiment. You just happen to get a snapshot, right? People that were working on Mount St. Helens before the eruption and it continued to work post the eruption, they're able to like really look at that natural experiment, right? We also can do natural experiments if we look at things like glacial retreat, or before and after a hurricane, or before and after a fire. Being able to observe those changes, what happens to a population, what happens to the environment, how the plants and animals recover, all of those things. Right. So usually a study is already ongoing. If like those are the lucky chances, right? I mean, my friend was kind of like when this fire happened, he was like, it's kind of like God looked down and was like, Corey, my boy, would you like to be on the cover of Science magazine? Like because it's a what like once in a generation experiment. Like you just happen to have all of the right data being recorded right before this thing happened, and so like you can't predict those. They're just like gifts from it's a divine intervention instead of demonic intrusion, right? It's just like this lucky thing that happens and you happen to grab it, huh? Right, just like sunshine, everything. Yeah. So more likely, instead of having like a before and after an event kind of thing, we look for natural variations. So you look for changes along a stream, you look for differences between two lakes that are really similar in a lot of ways but are separate. And so you can um, try to find something that has the variation you want to study naturally occurring. Okay. Okay. We can also think about these natural experiments in a couple of different ways. We can do a snapshot experiment. And so a snapshot experiment is replicated in space. So we look for um, variation that's occurring across space. And we go out and sample all that variation at the same time. So if we have 10 plots that vary slightly in some factor we're trying to measure, maybe it's elevation, right? Maybe it's berry growth. I don't know. We go out and measure all of those plots at the same moment in time, same season, same day, same temperature, and then we compare them. And so it's like a snapshot across space. And then trajectory experiment is a snapshot across time. And so you pick the same plots and you visit that exact same place year after year, season after season, whatever your time variable of interest is. Right. So the study out at Masson Ridge looking at vernal pools, visiting the same pools, applying the same methods to those pools year after year after year, and looking at how those pools vary over time. And so the trajectory experiments can help you to find trends, like what is the trend of change over time? Or is there a trend for there to be no change? Right? So this is a hodgepodge of types of experiments, right? And so we're probably going to do something that's more observational, but we can also participate in things like a trajectory experiment. I mean, Abigail's example of what's happening in the vernal pools is great. Very easily, somebody from this class could say, well, I want to go look at the vernal pools this spring, and I want to compare them to the data from last spring and the spring before, right? We have some existing data sets that you guys can work with. So things like the vernal pools at Masson Ridge or the redback salamander data in GCC's forest, right? We also have the potential for us to build some data sets, right? Sean was just talking about maybe we want to do an inventory of what are the different types of diseases in the forest. Maybe we want to look at species composition in our forest, right? And maybe we would come back in 10 years and do the same thing, right?
Levels of forest value die off.
Right, forest health and succession.
Succession, there you go.
Succession is what you're thinking
that's
of.
the word I was listening
Yep.
to.
And so,
Wasn't quite- Roman didn't wrong.
right.
Wasn't there.
Just because you guys can't do the entire trajectory in the time of this class doesn't mean you couldn't set up a trajectory experiment that students might have the potential to check back in on in the future. And so I'm always open to thinking about something like, it would be really interesting to know, like, what is the species composition of our forest, right? I have no idea. What percentage of our trees or what type, I don't know. It'd be really cool to know that now and know that 10 years from now, right? The student 10 years from now gets the cool update, but the description of it now is also really interesting, right? Things that happen on this campus are more interesting to me than things that happen somewhere else, mostly because we are trying to promote that one of the things that GCC has available is this option to work on campus and to learn about our campus. And we want to continue to build it as a learning resource. So the more we learn about it, the more we can make decisions about how we want to change things or how we want to build things out. At this point, we've got the gardens going. Right now, the big push is to work on the Meadow Farm garden in terms of our campus as a learning site. But the next step is the forest and doing some trail management and some invasive species removal. And so we as GCC, as the science department, are thinking about like how are we going to change our forest to use it in the future, right? And so any project that you guys want to do in that forest gives us information to include and think about when we move forward. So last year, Aisha did some looking in the forest for wetlands. I think that based on some data Jeremy gathered, in your Adopt a Plot project, looking at places where water was stagnant up there, and also another student reporting some things, there may be some places that could be breeding locations for spotted salamanders in our forest that we've just never assessed at the right time of year. And that would be great to know because we find juveniles in the forest. Where are they coming from? It'd be great to know where they're breeding. We did not find evidence for them breeding in the wetland. That doesn't mean they didn't breed there. We just didn't find evidence. We could go back and look at the wetland, right? So,
pretty easy in that wetland. Pretty easy to miss something.
Yeah, it's huge,
Yes.
right? So there's a lot of possible projects. We also have to be mindful of the time of year. And so, although it might be really interesting to want to look at things like insects and pollinators and flowers, they are going to just barely be getting started at the very end of our season, right? Whereas if we wanted to like sample plankton and microcrustaceans and things that are living in the water column in the wetland or in the duck pond or in the muck below the wetland, those are going to be there, right? It's hard to sample soil invertebrates right now when there's a foot of snow on the ground, right? They're gonna just still be waking up later in the season. We could categorize trees based on bark, but we can't look at overhead canopy, right? So just a matter of picking a question that's answerable in the time frame and that interests you. It does come in quick.
It's just like one day, it seems like one day you see really well in the forest, and the next day you're like, oh, why is it so dark?
Yeah, right? I can't see anything. Yeah. Right. Yep. So we're going to be doing most of our data collection starting at spring break. And I put that in there because that's about like, depending on what's going on with the year, usually around spring break is about when things like ice starts to melt off of the wetlands and the vernal pools. The snow stops being an overwhelming presence in the soil. There might be patches of snow, but the whole ground isn't under snow, right? And so that's about when we'll be able to start really sampling organisms. It's around spring break. So our plan from here forward is to spend some time thinking about what kind of projects we want to do. Next week, we're going to have our lab. I don't know if we're going, I'm going to send you an email. We might be going to the library. I think we're going to the library.
Yeah.
I don't think I confirmed whether Tim was coming here or we were coming to him. But we're going to work with Tim next week on developing research questions, which is a familiar activity for some of you. We're going to do it again. There'll be some things that are different than how you've learned it in the past. And if you haven't learned it before, it'll be all new. But we're going to work on spending most of next week figuring out what our research question is. And we're going to talk a little bit about how to write a research proposal. And then the next 2 weeks, February 19th and February 26th, so 2 weeks from now and 3 weeks from now, I'm going to give you some information on sampling methods and planning for statistical analysis and things that we can do. And then you're going to spend at least half of the class brainstorming with each other, working on your proposals, sitting and writing. Maybe go to the library and write a little bit, have some workshop time. And then our proposals are going to be due on March 5th. And so you'll actually submit a proposal for peer review on March 5th. So I'll give you those updated dates and stuff next week when we talk about writing the proposal. I'll give you a proposal assignment. I'm going to kind of put that on our radar though. We've got a bit of time to get a proposal written, But if you want to do the UMass Research Conference, we need to at least fudge an abstract much faster than that. Okay. Abstract's a summary. Yeah, a summary of your project. Yeah. Maybe make some connections. Yeah. Yeah. Right. So, yeah. So we want to think about moving that forward for you then. Get an abstract done. Gotta schmooze and make connections. It never hurts. Okay, I'm going to stop this recording. I was just going to record some of that update for people.